This study is an extension of the work that is ongoing in the lab since the mid-90s. We were the first lab to identify the PAR4 gene. This is a gene that was identified in cells that were undergoing apoptotic cell death. That's a natural way for cells to die, and this is a way that has been conserved in evolution. It's also the mechanism by which chemotherapeutic agents and radiation will cause shrinkage of tumors. So we asked whether this particular gene is selective in killing cancer cells, and our answer was that it indeed was. And one way to find out whether this gene selectively kills the cancer cells but not the normal cells, in fact that it's not toxic to the normal cells, is to make a mouse. And that's exactly what we did. We made a mouse. This is a transgenic mouse that was made in the transgenic facility here at UK, and this mouse is expressing the protein in all tissues of the mouse, and we asked whether this mouse is resistant to the growth of tumors, spontaneous tumors as well as inducible tumors. And our question was whether we could transfer the cancer resistance phenotype from the cancer resistant mouse to mice which are otherwise susceptible to the growth of tumors. And one way we did this was to transfer the bone marrow of the cancer resistant mouse to recipient animals which are otherwise susceptible, and we looked for transfer of that cancer killer activity in the serum of the recipient animals, and indeed we found that this activity was present in the serum. This activity was able to kill the cancer cells but not the normal cells, and the recipient animals were alive and healthy. The recipient animals did not show any indication of toxicity resulting from the expression of the protein, indicating that this protein can be very easily transferred from one mouse to the other. Where does it take us from here? We will ask whether we can genetically modify the stem cells of mice which are otherwise susceptible to cancer by injecting the DNA that's important to produce this protein, and then reintroduce those stem cells into the cancer sensitive mice. And then we can ask whether, first of all, these cells are able to grow and colonize the tissues of the cancer susceptible mouse, and then subsequently we can test whether the protein that is expected to be produced by the DNA will be available systemically in this mouse, and subsequently the study will ask whether the recipient animals are resistant to the growth of spontaneous tumors as well as tumors that are induced in these mice, either by crossing them with other mice or by asking whether metastatic tumors will grow in these animals or not. What this is really telling us is that by way of bone marrow transplantation, one can transfer the cancer resistant phenotype from one individual into another. In this case we've used only mice, but if I was to extrapolate this to what can be done in people, one can make genetically modified stem cells and introduce them into patients and look for the presence of cancer resistance. One of the advantages of introducing this particular gene is that this gene will not allow cancer cells to grow. So if we were to introduce this into stem cells, and for some reason the stem cells were to become modified and become aberrant in their growth characteristics, indicating that they might have become transformed or become malignant in their phenotype, this protein will stop that process and kill those stem cells. So that's really an advantage of using this particular gene to genetically modify the stem cells if one was to take this research further to apply it for the benefits of treating cancer patients.